Hello, my name is Michael Rivera, and today I'll be talking to you about your body's defense system, the human immune system.
Let's begin by looking at what your body is at war with: pathogens. A pathogen or infectious agent is a biological agent that causes disease or illness to its host. The term is most often used for agents that disrupt the normal physiology of a multicellular animal or plant, but can also attack unicellular organisms. There are many types of pathogens, such as bacteria, viruses, fungi, and parasites. Next, I'll break down your body's attack sequence. When a pathogen such as bacteria, virus, fungus, or parasite tries to infect your body, your first defense is your nonspecific external barriers. These are your skin and mucous barriers. Should the pathogen break the skin and enter your body, your innate immune response is activated. Your innate immune response is considered a broad spectrum attack. Should the pathogen be too strong for your innate immune response, the innate immune response will activate your adaptive immune response, which is a more specific attack tailored exactly for that specific pathogen. Your first line of defense is the external nonspecific barriers we discussed in the slide before. These barriers consist of skin and mucous membranes. The skin serves as a mechanical barrier to pathogens and that is tough to break and penetrate. Your skin also secretes lysozyme, an enzyme that helps in breaking down cell walls of bacteria. Your skin also maintains a pH between 4.0 and 7.0, which is considered mildly acidic to neutral. This acidic barrier helps with destroying pathogens as well. Mucous membranes are located in your respiratory and gastrointestinal and urinary genital tracts. These mucous membranes produce mucus that traps pathogens. You then rid the mucus of your body through your nose or throat. Your gastrointestinal tract also produces hydrochloric acid, which is a strong acid that helps in destruction of pathogens.
Now, we begin with the second line of defense, the innate immune response. The innate immune response is responsible for a variety of broad attacks. First is the initiation of inflammation. The process of acute inflammation is initiated by cells already present in all tissues, mainly resident macrophages, dendritic cells, and mast cells. These cells present receptors contained on the surface or within the cell named pattern recognition receptors, or PRRs, which recognize molecules that are broadly shared by pathogens but distinguishable from host molecules, collectively referred to as pathogen-associated molecular patterns, or PAMPs. On the onset of an infection, these cells undergo activation and release inflammatory mediators or cytokines responsible for the clinical signs of inflammation. The inflammatory response is characterized by the following symptoms: redness of skin, due to the locally increased blood circulation, which helps with the mobilization of leukocytes to the site of infection. Next, heat, or the increased local temperature, also known as a systemic fever, helps with increased mobility of leukocytes, enhanced leukocyte phagocytosis, and the decrease of endotoxin effects from bacteria or fungal infections. And then the overall swelling of affected tissues, which is due to the amount of leukocytes in the area. Another responsibility of the innate immune response is the recruitment of leukocytes, or white blood cells. There are a variety of leukocytes that help in the innate immune response. First, mast cells reside in connective tissue and in the mucous membranes. They are associated with wound healing and defense against pathogens, but are also often associated with allergic reaction and anaphylaxis. When activated, mast cells rapidly release characteristic granules or small secretory vesicles that are rich in histamine and heparin, along with chemokines or chemical cytokines that signal into the environment. Histamine dilates blood vessels, causing the characteristic signs of inflammation, and chemokines help in the recruitment of neutrophils and macrophages. Macrophages are large phagocytic leukocytes which are able to move outside of the vascular system by moving across the walls of capillary vessels and entering the areas between cells in pursuit of invading pathogens. Macrophages are the most efficient phagocytes and can phagocytose substantial numbers of bacteria and/or cells or microbes. The binding of bacterial molecules to receptors on the surface of a macrophage triggers it to engulf and destroy the bacteria through the generation of a respiratory burst, causing the release of reactive oxygen species. Pathogens also stimulate the macrophage to produce chemokines, which summons other cells to the site of infection. Granulocytes are neutrophils, eosinophils, and basophils. Granulocytes contain a variety of toxic substances that kill or inhibit growth of bacteria and fungi. Similar to macrophages, granulocytes attack pathogens by activating a respiratory burst. The main products of the respiratory burst are strong oxidizing agents, including hydrogen peroxide, free oxygen radicals, and hypochlorite. These substances, which disrupt the cell wall of the bacteria or fungi, helping create death. Dendritic cells are phagocytic cells present in the tissues that are in contact with the external environment, mainly the skin, and the inner mucosal lining of the nose, lungs, stomach, and intestines. They are named for their resemblance to neuronal dendrites, but dendritic cells are not connected to the nervous system. Dendritic cells are very important in the process of antigen presentation. Natural killer cells, or NK cells, are a component of the innate immune system that does not directly attack invading microbes. Rather, NK cells destroy compromised host cells such as tumor cells or virus-infected cells, recognizing such cells by a condition known as missing self. This term describes cells with abnormally low levels of a cell surface marker known as MHC1 or major histocompatibility complex 1, a situation that can arise in viral infections of host cells. Normal body cells are not recognized and attacked by NK cells because they express intact self MHC1 antigens. Another responsibility of the innate immune response is the responsibility for activation of the complement system. The complement system is a complex system of proteins that acts as a cascade. Each protein within the system is assigned a number and they react in sequence once the system has been activated. There are three types of pathways. First, the classical pathway is activated by the antibody part of the antigen-antibody complex. It can also be activated by enzymes and a variety of substances which include endotoxins, cell membranes, and viruses. The alternative pathway does not depend on the antigen-antibody reaction, but rather biological activators of this pathway, which can include bacterial endotoxins, yeast cell walls, aggregated immunoglobulins, also known as antibodies, and snake venom. The last pathway is the lectin pathway. The activation of the lectin pathway is dependent on the binding of a lectin to mannose of the surface of a pathogen. All three pathways are responsible for MAC, the membrane attack complex. The complex is inserted into the lipid bilayer, leading to movement of ions and water across the membrane and ultimately leading to cell lysis. One last responsibility of the innate immune system is the activation of acquired immune system, or response. Dendritic cells are antigen-presenting cells. Antigen-presenting cells are cells that display foreign antigens complex with major histocompatibility complexes or MHCs, as we stated earlier in the presentation, on their surface. Their main function is to process antigen material from the virus, bacteria, or fungi and present it on their cell surface to the T cells of the immune system. They act as the messengers between the innate and adaptive immune systems, thus becoming the link between both responses. The third line of defense is the adaptive immune response. It is responsible for cell-mediated immune response and the humoral immune response. First, cell-mediated immune response uses the T lymphocytes or T cells. T cells are produced in the thymus. Here, they are taught how to distinguish self from non-self antigens. They can recognize an almost limitless number of different antigens. Mature T cells are stored in secondary lymphoid organs, such as lymph nodes and the spleen. These cells circulate in the bloodstream and the lymphatic system. After they first encounter an infected or abnormal cell, they are activated and search for those particular cells. There are three different types of T cells. First, the cytotoxic or killer T cells attach to antigens on infected or abnormal cells. Killer T cells then kill these cells by inducing apoptosis, programmed cell death. The next T cell is helper T cells. They help other immune cells,
Some helper T cells help B cells produce antibodies against foreign antigens. Others help activate killer T cells to kill infected or abnormal cells, or help activate macrophages, enabling them to ingest infected or abnormal cells more efficiently. The last type of T cell is suppressor T cells, which produce substances that help end the immune response, or sometimes prevent certain harmful responses from occurring. The second part of the adaptive immune response is the humoral immune response. The humoral immune response uses B lymphocytes or B cells. B cells are formed in the bone marrow. They have particular sites on their surface where antigens can attach. B cells can learn to recognize an almost limitless number of different antigens, same as T cells. There are two different types of B cells. First, antibody secreting plasma cells produce antibodies that are specific to the antigen that stimulated their production. Antibodies attach to the antigen and form an immune complex, also known as the antibody-antigen complex. Antibodies protect the body in the following ways: helping cells ingest antigens, inactivating toxic substances produced by bacteria, attacking bacteria and viruses directly, activating the complement system, helping certain cells such as natural killer cells kill infected cells or cancer cells. The second type of B cell is B memory cells, which remember the specific antigen that stimulated production. Helper T cells help B cells in this process. The B cell response to antigens has two stages. The primary immune response is when B cells first encounter an antigen. The antigen attaches to a receptor stimulating the B cells. After their first encounter with an antigen, production of enough of the specific antibody takes several days, thus the primary immune response is slow. The secondary immune response is whenever B cells encounter the antigen again, memory B cells very rapidly recognize the antigen, multiply, change it to plasma cells, and produce antibodies. This response is quick and very effective. The diagram shows an antibody molecule. The molecule has two parts, the variable part and the constant part. The variable part varies from antibody to antibody depending on which antigen the antibody targets. The antigen attaches to the variable part. In conclusion, your body is a fascinating machine. Your body can create over 1 billion different antibodies that are tailored from the many different antigens presented by the many different pathogens. This is all done by the mixing and matching of your DNA. It also shows how connected life is from the smallest known organism in the world to the largest. And to know that this machine is working all on its own from your genes, that is a miracle.